Hey, good afternoon, everybody. Welcome to another Bio 20, Bio 20? Bio 4B lab. And in this lab, what we're going to do is we're going to do some water quality and hand washing bacteriology on here. So the very first day that we actually week that we would be in lab, go over some of the lab safety rules and so forth. And first thing we'll set you guys up is do the hand washing bacteriology setup. And because this actually takes a couple of days for us to actually get this lab done, because we need to allow for the samples to incubate. And then on your way home this very first day, you will actually get a water sample or a water container to go collect some water samples from an outdoor location of your choice. So it could be a creek, it could be a lake, it could be a pond, it could be some waterway around your house, it could be the ocean. And so then you'll bring that back. I've had students go to Disneyland, although nobody's going to Disneyland right now, Pirates of the Caribbean. So that's what we're going to do is we're going to take a look at some prokaryotes, more specifically bacteria, in this particular lab. So we're going to start out with the hand washing, and then I'm going to walk you guys through that, and then we're going to do the water quality next. So in this portion, I'm going to go ahead and just briefly talk about what you're actually going to be doing, and then I'll show you guys the video of actually what I did, and then we'll come back and we'll talk about the results from that. So you're going to get a Petri dish with nutrient agar in there, and you're going to divide this into 4 quadrants, and you're going to number them 1 through 4. And as you do that, in quadrant number 1, you're going to place your fingertips on there. This is with your unwashed hand. Then what you're going to do is you're going to wash your hands like you normally would. That's what I typically tell the students. Wash your hands like you normally would. So if you only wash it really quickly, don't spend a lot of time. Just do what you normally do when you wash your hands. If you don't use soap, don't use soap. If you use soap, use soap. If you use cold water, use cold water. If you use warm water, use warm water, and so forth. All right, and then afterwards, dry your hands. Maybe don't dry your hands. Maybe just kind of, all right, or you wipe them on your clothing. I wipe it on my clothing a lot because I know my clothing is clean, and so especially when there's no paper towels and so forth. Okay, and then once we have all 4 fingerprints done, and then we're going to repeat this 3 more times, and then we're going to incubate for 24 to 48 hours depending on what day of the week it is and when we're able to come back into class. Okay, so what we're going to do right now for Bio 4B is we're going to do the hand washing technique, and then we're going to look to see how much bacteria can be found on your fingers. Now with this, I'm going to do this twice. I'm going to do this today, and then I'm going to do this tomorrow when I come back in here. Today I've actually been washing my hands a lot, so I'm not sure how the results are going to turn out. But I know the last time I did this back in January of 2020 on my initial one, because I was rubbing the tables and everything, I was touching a bunch of stuff in the lab, and on my first set of prints, I actually got some fungus growing. I wasn't the only one that actually got fungus on there. So here's what I'm going to do. This is a nutrient agar, and I actually divided this thing up into quadrants. So I got 1, 2, 3, and 4. So what I'm going to do is I'm going to put my initial unwashed hands, fingertips, on these 2 spots, on spot number 1. Then I'm going to wash my hands like I would normally wash my hands, and then I'm going to place it here, wash them again, place it there, wash it again, place it right there, and then this is going to go into the actual incubator. Okay, and then I'll do this again with this Petri dish, but I'll do this tomorrow when I first get in, so then that way I don't have as clean of a hands. All right, so hopefully, but I have been touching a couple of things. I've been cleaning up the lab right now. So here's what I'm going to do. Okay, so the Petri dish is always upside down because you can see the condensation down here. All right, and that's to prevent the condensation from dripping onto the actual specimen. And you always want to keep this. You always want to keep the Petri dish down so that it doesn't allow for other things to fall on there. All right, so all right, so you can see my finger. I don't know if you guys can see it, but might be able to see my fingerprints right there. Now that I'm actually breathing on there. So this is going to go off into the incubator, and we'll see you guys tomorrow. So I pulled this out of the incubator. It's been in there for 24 hours. And what I'm going to do is I tried to, I haven't really washed my hands a whole lot. I did kind of getting cleaned up, but I've been trying to touch a bunch of stuff, you know, and just to see, you know, how much bacteria I could potentially pick up. But what I wanted to show you guys is, this is from yesterday, where I actually, before I did this part of the experiment, I actually was washing my hands a lot. And as I do that, you kind of can't see as much, but as I flip this over, and you kind of hard to see under this light right over here, but I'm going to place it right here on the dark aspect of it. And as I move a little closer, I think you guys should be able, there we go. I think you guys can see some of the dots on there, like right over here and right over here. So that's number 1 and that's number 2 right here. But if you notice, number 3 and number 4 is actually, they're actually pretty clean. Typically what well, there's a little bit of spot, I see a little bit of spots in 3. There might be something in 4 also. Okay, and so what we're going to do with that is we're going to gram save, but I'm going to save that until we get the sample from over here to see if we get anything kind of different. So I'll do gram staining all together at the same time, not today. But what I'm going to do next is I want to take this, we want to head over here to the sink, and we're going to do the same hand washing aspect of it. So this is a clean one, but I haven't really, I've been no gloves, and so I just kind of been touching stuff. All right, so here's the, so I'm gonna just rub the countertop a little bit and see what else we can pick up. So again, I'm going to place my 2 fingers in spot number 1. All right, and then I'm going to there we go. All right, so I'm gonna wash my hands one last time, and then I'm gonna put that into the incubator for 24 hours, and we'll take a look at it tomorrow. So after watching the demonstration video portion, and you saw where after the first day of me washing my hands, and I've actually washed my hands a whole lot, so I had to redo this particular experiment. At least I wanted to. So here's day 1. After 24 hours, you can see some spots over here in number 1 and number 2, but you don't really see anything in 3 and 4. And so typically, as you wash your hands more, you'll get more of the bacteria out of your fingers, your crevices, and so forth. So I let this go for 1 more day, and so this is the result after incubating for 48 hours. So you can see where number 1, actually you can see where number 1's growing in spot number 2, right, where we can see right here, and then spot number 3. Right, we got more over there, although you can't really see anything over here. And then spot number 4, right, we can actually see a little bit more. And just to kind of reiterate, when I did this in January 2020, myself and several other students actually had some fungus growing in spot number 1. And I know, because often I'm rubbing the tables and so forth. So I came back and the day that we looked at this one, so this will be day number 2, right? So what I did was I did not wash my hands when I came in that day, and I touched a whole bunch of different things, and then I just proceeded like I did before, like you heard, and notice that spot number 1, look at spot number 2, 3, and number 4. So after this, what we're going to do now is we want to gram stain to see what kind of bacteria that we have. Is it gram positive? Is it gram negative? Are they bacilli? Are they cocci? Are there spirillum on there? So what I did was I pulled one of these colonies off of each one. So I did a gram stain for each one of those, and I pulled the colony off, and I used aseptic technique to place it onto a microscope slide. So I'm going to go ahead and go over the gram staining results right here. But the gram staining, I did this at the same time when I did the water quality stain. So, and it's actually going to be in the water quality video where I'm going to explain to you guys how to gram stain. Okay, and so with this part, what we're looking at is these are the two samples that I had from RSM Lake. And one of the things that, I'm sorry, RSM Lake. Yeah, yeah, I discombobulated. So these are my day 1 hand washing, day 2 hand washing. Okay, so and so if you notice, hopefully you should be able to see what do we have right here, right? We have gram-positive Staphylococcus on both of them, and so this is part of our normal body flora on there. So I didn't find anything else on there that was out of the ordinary. So this is most likely my resident Staphylococcus that I have on my skin. As we take a look at the results, right, why did we get what we got? And so I was expecting gram-positive Staphylococcus aureus because that's part of our normal body flora. But you notice that as we wash our hands more, and that's the one big thing that we really noticed back in January 2020 when we were doing this lab, a lot of the students, when they got to quadrant 4, had a whole bunch more colonies on there. And part of the reason for that is as you wash your hands, you're getting more of the bacteria out of your fingertip ridges and grooves, right? So here you can look at your skin, and then, you know, at the base of your hair follicles, you know, where you have your sweat glands, right? And so here is a sebaceous gland. So this is an oil gland right here. And then this is a sweat gland. So in these areas, you're going to have some bacteria. So as you wash your hands more, you're going to be lifting them out. The other thing that we can kind of look at is what's your skin condition like? Are you, how acidic are you? Our skin's slightly on the acidic to retard some of the growth of some of the microbes on there. You know, do you have dry skin? Do you have oily skin? Did you put lotion on, you know, beforehand? You know, what did you have on your hands? What were you touching and so forth? So all those skin conditions will actually affect what we have. So if you're looking at our skin, right? So this is normal skin, nice and smooth. You can look at our skin right through here, and it can be pretty darn wrinkled on there as well. All right, so when you look at these, especially like this fingertip, look at these deep grooves that they have. So what we're looking at is something that looks like that. Not necessarily wrinkled, but we got a lot of these grooves on there. So the bacteria that I was pulling off of there was part of my normal body flora. So I was expecting Staphylococcus on there, gram-positive Staphylococcus, right? That's part of our normal body flora. They're there to defend us. They're there to produce antibiotics to help prevent other transient bacteria from coming aboard. So you can see the second term right here, transient microbiota. What is transient microbiota? This is the biota that's there short term. Your normal microbiota or your resident flora, that's there all the time. So now you touch a surface, you touch a doorknob, you touch a countertop, you touch somebody else, you shake somebody's hand, you give somebody a kiss, okay? You give somebody a hug, you do all these different things, and you're going to pick up transient microbiota. And those are things that are there temporarily, right? And so one of the things that has occurred in court. I had one of my former students that was a bouncer at a bar. He got into a fight, and one guy claimed that he punched him, right, and knocked him out and broke his jaw or something like that. And he claimed, I didn't touch him. I hit the other dude, but I didn't touch this guy. And so what they actually had done was, because he had cuts on his hand early on, they took a swab of that, and they were actually able to say, we have data to support that this bacteria that we found on the bouncer's knuckles does not match the bacteria that's found from that said individual. So he was like, whew, thank goodness, I told you. So, but here's the thing, the more you wash your hands, this is why you're supposed to sing the alphabet song, all right? This is why you're supposed to wash with warm water and soap, right? And back in spring 2020, actually some students, they don't really wash hands. So a couple of students actually used hand sanitizer, and I'm trying to remember what the results were like, and I just can't quite remember what they actually had. I don't think it was less. It was pretty consistent on there. Okay, and plus you got to remember, hand sanitizer, you're supposed to leave it wet for like how many minutes? Like 2 to 5 minutes on there to allow for the antibiotic, the antiseptic on the alcohol wipes to work. If you have any questions on this, let me know, because you guys are going to have to identify gram-positive, gram-negative bacteria and the morphologies on your lab exam, or on the quiz first, actually. All right, everybody, welcome back again. So here is the water quality bacteriology portion of this lab. And in this lab, what I did was I went out to 2 lakes. Well, I didn't go out to both lakes. I went to one lake, and my daughter's boyfriend, who was working at Lake Mission Viejo, collected some samples for me from there. And so this was back in July 23rd and July 25th of 2020. He went out to Lake Mission Viejo. He went to the boat rental dock area, and he collected about 250 mils of Lake Mission Viejo water for me, and a small, like a 10 fluid ounce naked juice bottle that was cleaned out. Okay, and then I went to Santa Margarita Lake on July 25th and collected from this spot right over here. So this is the amphitheater. So we're, what is it, Las Flores? It is right behind me, and then Santa Margarita Parkway is way over there. So I actually went right here to that location by the amphitheater, and I collected the same volume of water from there. And both water samples were placed in a Ziploc bag just in case it spilled over, and that's one of the biggest warnings I always tell you guys, is put it in a Ziploc bag, because I've had students, even though I warned them, they forgot to do that, and it was in the back seat, it tipped over, and then their car stank. And plus, you don't want to leave it in the car, because then that allows for the bacteria to continue to grow. So put it in the fridge, and I brought it into the lab on July 27th. And that's the day when I began the water quality testing on there. For the water quality testing, we're going to do the most probable number determination based upon the water sample that you collected. We're going to do a series of serial dilutions into a lactose fermentation tube that has bromothymol blue chemical indicator in there, so that's a pH indicator. And inside, to test to see if we have gas, is going to be an inverted Durham tube. So you can see this tube right here is actually trapping some gas right there. But here's the inverted tube. Here you can see the bromothymol blue, and inside there's lactose. And so what we're going to do is we're going to have 3 triple strengths, 6 single strengths, and then to each one of these set of 3 tubes, we're going to put varying amounts of our sample. So for example, from our original water sample that you collected, we're going to put 10 mils into each of the triple strengths. We're then going to put 1 mil into 3 of the single strengths. We're going to put 0.1 mils of our sample into the last 3 single strength tubes on there. From there, we're going to incubate this for 24 to 48 hours. So depending upon what day it is, it might actually go over the weekend. And so it might be a lot longer. And so with this, what we're going to do is we're going to look to see if we have fermentation, do we have gas production, do we have, how many of the tubes and so forth. And then we're going to use this chart right here, which you can't actually see. So after about 24 to 48 hours, we'll come back and we'll look at the tubes. So this one, you can see 3 positives, 2 positives and 0, so I can't see this because that's so small. If I put my glasses on, let's see. Heck no. Okay, I can see a little bit. So there's 3. We're going to come down here, but we need to find 2. So there's 2, 3, 2, 0, and dang it, I can't read that. I think that says 93. And so let's say we have 93 microorganisms per, it's supposed to be per 100 mils, but we actually have per 250 mils, thereabouts. Okay. And so from one of the 10% positive tubes, then what we're going to do is, oops, I forgot, we're going to do what's called the confirm test. And the confirm test is we're going to then put it onto this EMB plate. EMB is a selective medium, and that is eosin methylene blue. That's what that's what the EMB stands for. It contains lactose and sucrose, and so we're going to look to see if we get fermentation again, because we're going to get a color change. And so the chemical indicators on here is going to, the eosin and the methylene blue, they're going to interact, and they're going to give us a pH indicator. Deep purple, meaning, and with a metallic green is a strong acidic solution, and you can actually see the metallic green right here. So one of the things that tells you, that's a key indication of, is that you have E. coli. All right, so there's some gut bacteria right there. Now the ones that are non lactose fermenters, you'll get a pink or translucent color. That's what you'll see right here. From that, we're then going to take one of these isolated colonies over here, and we're going to put it into another single strength fermentation tube, and we're going to do the completed test, because we want to know if that isolated colony, is it the fermenter, is it the gas producer, does it do both, does it do neither of those? So like over here, this will be a negative for fermentation, but a positive for gas production. Then we'll also take another one of these isolated colonies, and we'll put it onto nutrient agar slant, and then that way we can gram stain after that. And so, but these samples now will go into the incubator for another 24 to 48 hours. So a close-up view of the presumptive test. So we have our water samples, right? And then we're going to, you're going to see in the video where I actually transfer all this across, and we're going to have 10 mils in the triple strength. We're going to have 1 mil in a single strength, and then we're going to have 0.1 mils in the single strength. So here's the RSM lake. Here is Lake Mission Viejo on there. Off to the incubator. Come back after the incubator. We're going to look for a positive and negative fermentation and gas production. So over here on this side, you can see this is a negative for gas and negative for fermentation because it's still green. This one right here on this is happen to be this triple strength. You can tell by the sheer volume in there. Positive for fermentation, you can see positive for gas production right there. And then this is a single strength, you got positive for gas production right here and positive for fermentation. And so based upon this, now we can actually see this chart a little bit better. So if we go back to that one example, it was 320, so 320, oh, 93. Whew, I could still pass that eye chart exam. So the NP index says per 100 mils, but we actually had about 250 mils. I need to really take an actual measurement on that one. All right, so we collect the water samples from RSM Lake and Mission Viejo Lake. Okay, so RSM was collected on 7/25/2020, and Mission Viejo Lake was collected on 7/23/2020. Both have been in the refrigerator to prevent additional bacterial growth. To this, we actually have, for each water quality, we're doing what's called the presumptive test, and we're going to have 3 triple strengths. So you can see the black labels right here for Mission Viejo and for RSM Lake right here. So RSM and Mission Viejo. Okay. To that, we are also going to have the single strength. We're going to have 6 of these. So you can see 6 right here, 3 and 3, 3 and 3. Okay, so there. To each one of these, we're going to add different volumes of our water sample. On the triple strength, we're going to add 10 mils. Right, so we're going to use this disposable pipette right here, and we're going to put in 10 mils of each into each one of these. And then on the single strength, we're going to put 1 mil. I'm going to use a P1000, right, to put in 1 mil, and then I'm going to use the P200 to put in 0.1 mil. So 1 mil and 0.1 mils for each one of those. Cap this back up, turn the gas off, and I'll go ahead and unplug that. Okay, so we're done with this part. So now these actually have to go into the refrigerator, into the incubator, and this will be in there for 24 hours. So come on, we're going to go over here to the other classroom. So here you can see 37 degrees. We'll leave that in there for 24 hours. Here are the tubes for the presumptive test. We have the RSM tubes right here. So you can see how we have some fermentation that's taking place. Okay. So here, this tube right here is yellow in color from our original green. And this demonstrates that we do have fermentation. And then if you look right here, Mission Viejo on the other hand, so here are my Mission Viejo samples. You can see we have a little fermentation on the 3 10 mils, and there's a little bit of gas production right there, just a little bit, not a whole lot. So we have 3 right there. So here are the results for Rancho Santa Margarita Lake after 24 hours of incubation. And one of the things that you can see here are my 3 triple strengths for 10 mils, 3 single strengths for 1 mil, 3 single strengths for 0.1 mils of sample that's been placed. So if you're looking at this, you can see yellow, that is positive for fermentation. And so the 10 mils, I got gas and I got fermentation on there, so check. All right, so there's 3 of those. For the 1 mil, the single strength, I got fermentation for 3 and I got gas for 3. So, all right, so I'm good. So I'm 3 there. But this one, I got no gas. You know, that's a little bit bubbles. That's really not much, right? So no gas, no fermentation. So then I have 0 positive tubes over here. And so we come over here and then we look at 0 for 1, for the 0.1 mils, 3 for the 1 mil, and 3 for the triple strength. So we end up with 240, right, microorganisms per 100 mils, microbes per 100 mils. So that's the results for RSM Lake. So that's not too bad, even though we got E. coli in there. And oftentimes, a lot of times, I get samples from students that are way down here. For Lake Mission Viejo, the results are, so again, you can see our still our same setup. Yeah, you know, it's when we take a look at this, the 10 mils, the 1 mil, the 0.1 mil. So you look right there. There's, you know, it's kind of questionable. You called it slightly fermentation on there. I probably could have said 0, but I went ahead and called those 3, and those are 0 there, those are 0 right there, so we end up with 0, 0, 3, so we end up with 23 microbes per 100 mils. And so one of the things here that we're looking at, I mean, we could end up with 0, 0, 0, right? So then our MPN index would be less than 3 microorganisms per 100 mils on there. And so notice the big difference between Lake Mission Viejo and RSM Lake. So in this changes from season to season and from year to year, because I've gotten a whole variety of different results from this when I think back about it. So after the presumptive test, we're going to take one of our tubes that has at least 10% gas production and fermentation on there, and we're going to flame a loop right here, and we're going to allow that loop to cool, and we're going to take a loop full of that sample, and we're going to place it into, we're going to streak plate it. So in between, when I streak plate it, I might not be doing it correctly, but I streak it and I put it back upside down. You can see how this Petri dish is upside down to prevent condensation from dripping down into or onto the agar and where our sample is. So I'll put that down and I'll flame it again. I'll allow the loop to cool, and then I'll streak it by grabbing the tail end of where my first sample was. So I rotate it like quarter turn, and I streak it. I'll flame it again. I'll quarter turn it, streak it again, flame it, cool, streak it again 4 times to isolate. I'm not coloring it in because I want to find isolated. We're going to do the confirm test, and with the confirm test, that requires the use of a selective media. This is called EMB. And this particular type of agar is selective for typically gram-negative bacteria. And what we're going to do is we're going to take our tubes. We're going to take a tube that has at least, has gas production, at least 10% gas production. So we're going to take a positive tube. And what I'm going to do is I'm going to streak plate it so that we can isolate the colonies. And then once we have the colonies isolated, we can grow it up on a nutrient agar slant. And then we can actually kind of gram stain it as well. So we'll do gram staining in a couple of days. We'll do all the gram staining all at one time as opposed to doing them at separate times. All right, so okay, so I'm going to take this, streak it, I'm going to flame this so you can see I spilled a little drop of water right there. Okay, and then what I'm going to do is I'm going to drag, so you can see, I don't know if you can see that part. Turn that off. Okay, so these now are ready to go back into the incubator, and we're going to take a look at those tomorrow and after 24 hours. So after 24 hours, we're going to come back and we're going to check our sample. So here you can actually see, here's my initial streak, and then I flamed it, and then I streaked it again over here the second time. And now you can start to see isolated colonies. I didn't dip it back in there. Right. And then here's my third one, and you can't really see anything on my fourth one over here. And then, so here's RSM Lake. You can see we got dark purple, so we have acid fermentation. We got metallic green. So what do we have right there? We have E. coli. And so over here on this side, here is Lake Mission Viejo. So remember, the EMV plate, right, the methylene blue is actually going to inhibit most of the gram positives. But if we leave it long enough, we could get some gram positives in this. But we're going to double check because we're going to do gram stain also from this. And so you can see this is pretty translucent, maybe a little bit pink. I know it's a color image. I mean, it was way more metallic green when you're actually looking at it with the naked eye on there. So negative for E. coli over here, positive for E. coli over here, metallic green sheen. So make sure you understand that because you'll see that on a lab exam. So after the confirmed test, we're going to do the completed test. So to do the completed test, I'm going to flame a loop, and then I am going to pull one of these isolated colonies off of there. I'm going to place that into a single strength fermentation tube. We want to know if that's the colony that is the gas producer, is that the fermenter, does that do both, or does that do neither? And then what I'm going to also do is I'm going to flame the loop again, and then I'm going to get another colony that looks exactly the same, and then I'm going to put it onto a nutrient agar plate, nutrient agar slant. And I'm going to allow that to grow for 24 hours, and from there we can actually gram stain. And sometimes, oftentimes, I'll actually just gram stain straight from here, but it might kind of muddle some of the things. But for the most part, it does a pretty good job. We can actually do a pretty good job from here, but this is just in case, so we can actually do that. Lactose fermentation tube, and we want to double check and make to see if the organism that's growing on these plates, if they are the fermenter, right? So remember, with fermentation, that'll cause it to turn to this yellow color. And then if it's a gas producer, here you can actually see the gas a little bit better right here, right? So here you can see the gas production. And with the gas production, you can we want to double check to see if the organism, are they the fermenter, are they the gas producer, do they do both, do they do one, or do they do neither of those? Okay, so that's what we're going to do with the completed test. Incubate this for 24 hours, and then we'll double check those again. And at the same time, what we're going to do is we're also going to gram stain. And when I do the gram stain, I'm also going to do the fingerprinting one together at the same time. All right, so for the confirmed test, all right, what we're going to do here is we're going to take a look at RSM first. And if you look at RSM, whew, the smell. So this is that selective media. I think you guys can get a closer look right here. All right, see that metallic green sheen right there? That tells you that there is E. coli in the water sample. So if you're looking on there, there's a couple of different, looks like there's a couple of species on there. So you can see the little small isolated colonies on there. All right, and that's what we're going to do. So I am going to flame this loop right here. Get this nice and hot. And I've already labeled my sample right here. All right, so this is for whew, this one does smell. Too bad you guys don't have the smell-o-vision. So to do the gram staining now, I'm going to flame the loop again. I'm going to pull one of these individuals off of here, and then I'm going to have a drop of water, a little teeny tiny drop of water, and then I'm going to kind of smear that sucker around. I'm not going to do it like I'm buttering toast or anything, but I'm going to place it right in there. I'm going to allow that to air dry, and now go through a series of other steps on there. Or we can actually do it from the nutrient agar slant as well. So to gram stain, once this is air dried, here you can see the varying chemicals that we're going to add. We're going to add grams crystal violet. We're going to add grams iodine. We're going to use some acetone alcohol, and then we're going to lastly stain it with grams safranin on there. So here you can actually see, the staining process, and then we're going to look at it underneath the microscope to see what bacteria that we have. And so notice that this is on the pink side. It is not that really dark purple. So we're looking at gram negative bacillus on there. And then for the one that you guys saw earlier, it's E. coli. So all of our samples have dried. So here are the two for the fingerprints. So day 1, day 2. These are my RSM Lake one. So this is the E. coli. This is the other little purple one that's over there. Again, all these should ultimately be gram negative because of the EMB plate. And then this is the one from Mission Viejo. So what I'm going to do is, what I need to do next is I need to heat fix these. And to heat fix this, what you do is you pass this over. Now, students in the past, what they've done is they will hold the slide this way, but then the flame hits the bottom and it puts the flame up on the finger. So you can heat pass it where it's up this way, and you don't need to hold it there, right? And then if you're worried about the flame, you got a little clothespin, and all we're doing is just passing it through a couple of times. So now to do the gram stain, we're going to use the gram crystal violet, all right? And this is if what happened is this on the cell wall. So hopefully you've already remembered about the peptidoglycan layer. So on gram-positive bacteria, that peptidoglycan layer is really thick. But on gram-negative bacteria, it's a lot thinner. But on the outside of the gram-negative bacteria, they have the lipopolysaccharide. There's another membrane on the outside. So the crystal violet may not necessarily pass through there. So what I'm going to do here is I'm going to flood this. I'm going to leave it on there for 20 seconds. All right. I'm going to do the same thing for these guys over here as well. Do all this at the same time. So after 20 seconds, what I'm going to do here is I'm just going to tip it on the side. And then I'm going to rinse off the excess. So then after that, we're going to flood this with gram iodine for 1 minute. Okay, so we're going to leave this on here for 1 minute. And then after 1 minute, what we're going to do after 1 minute is we're going to use the acetone alcohol to kind of decolor it. So this should be 1 minute here for these guys. So now what we're going to do is we're going to take a look at it underneath the microscope, and we're going to see what the morphology is and what type of bacteria that we actually have. So that's gram staining right there. All right, so now we got to get, and now we got to dispose of that properly on there. So with the completed test after 24 hours, Lake Mission Viejo, I actually had 2 tubes because there was two different colonies. They were probably actually the same, but I grabbed 2 of them, and so they were both positive for fermentation and for gas production. The Lake Mission Viejo now is negative for both. Did I make a mistake earlier? Oh well. So that's the result after the completed test. Now what you're going to do is you're going to take a look at the water quality based upon the results that you have. Here is your actual assignment. You're going to write a 1-page paper, up to a 1-page paper, and these are the topics that you're going to address. So you're going to write this to, like you're submitting a report for the local city government agent, local authority, to whether or not they should open up this particular area to allow for people to use it. What if some people want to drink it? What if they want to eat fish out of it? I don't know. Whichever. You guys decide on that. If you have any questions, shoot me an email, stop in the Zoom office hours, swing by, let me know, and I'll get you straightened out with that. All right? So if you got any questions, what are you going to do? Get a hold of me. I'll talk to you guys soon. You guys have a great day. Hydrate. Stay cool. Talk to you soon. Bye.